Welcome, curious minds, to the deep dive. Today, we're embarking on, well, a monumental journey. We're not just glancing at the human nervous system. We are diving deep, aiming for a true masterclass. This isn't about, rote memorization, but really grasping the profound insights that allow professionals in this field to truly understand the architecture of thought, sensation, and movement. We're going beyond the surface, exploring every intricate detail to achieve a truly comprehensive, almost elite professional level of understanding. Okay, let's unpack this incredible system, piece by intricate piece.
And this journey is truly captivating because it reveals how neuroanatomy, the study of structure, and neuroscience, a study of function, aren't just academic disciplines. What's fascinating here is how these two fields, neuroanatomy and neuroscience, converge to answer the most profound questions about human experience. From the simplest sensory input, feeling the warmth of the sun, to the most intricate higher cognitive functions like dreaming or composing a symphony. Well, these fields work in concert to unravel it all. It's an immense challenge, sure, but the rewards of understanding are truly profound.
Absolutely.
Yeah.
So our mission today is to thoroughly dissect the nervous system. We'll trace its incredible journey from embryonic development and fundamental structural organization right down to its intricate physiological functions, specialized sensory systems, and even its vital vascular supply. We'll draw insights from foundational texts like Gray's Anatomy for Students and explore every concept to give you a comprehensive understanding that feels both deep and immediately relevant. All right, let's start with the grand blueprint, the foundational division that organizes this entire sprawling system. Imagine it as having two main operational branches.
Exactly. Structurally, the human nervous system is fundamentally divided into the central nervous system, or CNS, and the peripheral nervous system, the PNS. Think of the CNS as your body's highly protected command and processing center.
Right.
It comprises the brain and the spinal cord, our most vital organs. These are not just housed, but actively safe.
So that's your protected core processing unit, like a sophisticated main computer.
Precisely. And then you have the PNS, which is this incredibly expansive network reaching out from that central command. It includes all your cranial nerves that emerge directly from the brain, your spinal nerves branching off the spinal cord, and the intricate networks of autonomic nerves regulating internal functions, along with the enteric nervous system, often dubbed your second brain in the gut.
Wow.
The PNS truly extends throughout your entire body, connecting every muscle, gland, and sensory receptor back to the CNS. It's the communication infrastructure.
So it's a beautifully organized system, a securely housed command center, and then a vast, far-reaching network. Ensures every part of your body is connected and responsive. It's a fundamental structural division that underpins everything else we'll discuss. Now, how does something this complex even begin to form? Here's where it gets really interesting. The incredible embryonic journey that gives rise to this entire system. Starting from just a simple layer of cells, it's truly a marvel of biological engineering from the ground up.
It is, and it happens remarkably quickly. During just the third week of embryonic development, the outermost layer of the embryo, the ectoderm, begins a crucial transformation. It thickens to form what we call the neural plate, essentially a flat sheet of cells with immense potential.
A flat sheet that contains the entire future nervous system.
Exactly. This plate then doesn't stay flat. It develops a neural groove, kind of like a valley forming down its center, flanked by rising neural folds on either side. These folds continue to deepen and eventually fuse together. This pivotal process, called neurulation, creates the neural tube, a hollow tube-like structure with an inner neural canal.
Okay.
This fusion starts roughly at the midpoint of the embryo and extends rapidly, both cranially towards where the head will be and caudally toward the future tail. The entire process is usually completed by the fourth week.
So a simple plate folds into a tube within a week, and that tube then begins to sculpt itself into the distinct parts of the brain.
That's right. As cells continue to proliferate rapidly at the cephalic or head end of the neural tube, it undergoes dilations, forming the three primary brain vesicles, the very first recognizable signs of a brain. The prosencephalon will develop into the forebrain, responsible for our higher cognitive functions. The mesencephalon is destined to become the midbrain, a crucial central processing hub. And finally, the rhomencephalon develops into the hindbrain, forming the more posterior brain regions.
And the rest of the tube.
Caudally, the neural tube lengthens and narrows, becoming the spinal cord. Importantly, the neural canal within this tube doesn't simply disappear. It forms the cavities of what will become the brain's ventricular system, housing cerebrospinal fluid and the central canal of the spinal cord.
So that initial hollow space is maintained and evolves into the brain's intricate internal fluid system, providing both support and a transport medium.
Precisely. And it's not just the CNS forming from the neural tube. The peripheral nervous system also takes shape. It's formed by nerve fibers extending from the CNS and by neurons and their fibers that develop from migratory neural crest cells.
Neural crest cells, right? They're fascinating.
They are. These neural crest cells also originate from the surface ectoderm, much like the neural tube itself, initially lying adjacent to the developing CNS before migrating throughout the body to form diverse structures, including much of the PNS.
This profound journey, from a simple ectodermal plate to such a complex, functionally diverse nervous system in just a few weeks, it's a stunning example of precision biology. It really highlights the incredible efficiency and genetic programming that orchestrates our very foundation. Navigating the nervous system, with all its twists and turns, requires a very specific language for location and direction. Imagine trying to navigate a new, incredibly complex city without a clear map or consistent street names. That's what studying neuroanatomy without these specialized terms would be like.
You've hit on a crucial point. For simpler, linear nervous systems like those in fish, orientation terms are straightforward. Ventral, from belly, points towards the ground. Dorsal, for back, points towards the sky. Rostral, from beak, points towards the head. And caudal, meaning tail, points towards the tail. It's a simple, continuous front-to-back, top-to-bottom reference.
That makes a perfect intuitive sense for a four-legged or aquatic creature.
However, humans, being bipedal and having an erect posture, introduce a fascinating anatomical wrinkle. Our nervous system actually makes an obligatory bend, about 80 to 90 degrees, right at the midbrain diencephalic junction.
Ah, the bend.
Yes. Because of this bend, those classic directional terms, ventral, dorsal, rostral, and caudal, actually have different meanings depending on whether you're looking at the brain above the bend or the spinal cord below it. For example, your cerebral cortex's rostral is pointing forward, but your spinal cord's rostral is pointing upward towards your head.
So it's not a straightforward north, south, east, west anymore once you hit that bend in the midbrain. It sounds like we need a more universal set of terms that are consistent throughout the entire axis.
Exactly. To maintain consistency, we use additional terms that keep their reference constant, regardless of the bend: anterior, posterior, superior, and inferior. These terms maintain their meaning whether you're describing the brain or the spinal cord. Furthermore, to study the nervous system, whether through imaging or in the lab, we often examine it in sections. These sections are typically cut along one of three standard planes.
The classic slices we see in every anatomy book.
Precisely. First, the coronal plane divides the nervous system into anterior and posterior parts. Think of it like slicing a loaf of bread from front to back. Second, the sagittal plane divides it into left and right parts. If it's precisely at the midline, it's a mid-sagittal section. If it's off to the side, it's parasagittal. And finally, the horizontal plane, also known as axial or transverse, divides the nervous system into superior and inferior parts. Understanding these consistent planes and terms is absolutely foundational for any precise discussion or diagnosis in neuroanatomy.
It's a vivid reminder that even our evolutionary shift to an upright posture has fundamentally reshaped how our brain is organized and how we talk about it. These specific terms are truly essential for any precise understanding. So we've talked about the grand blueprint. Now let's zoom into the microscopic workforce. What are the actual workhorses of this system? The tiny, incredible cells making it all happen at the most fundamental level.
At the cellular core, the nervous system is primarily composed of two main types of cells: neurons, which are the nerve cells, and glial cells. The incredibly complex neurochemical signaling that underpins everything we think, feel, and do is predominantly carried out through the physiological connections between adjoining neurons. They are the communicators, the information processors.
And what does a typical neuron look like? I know they're incredibly diverse, but what are the core components?
Well, while neural morphology is indeed highly variable, a typical neuron has three main parts. First, the cell body, or soma, which contains the nucleus and is responsible for synthesizing macromolecules and integrating electrical signals. This is the neuron's command center.
Okay.
Second, you have dendrites, short tapered extensions that act like antennas, primarily receiving input from other neurons. Finally, there's the axon, a long cylindrical process that conducts electrical signals away from the cell body to other neurons. At the end of the axon are axon terminals, or synaptic endings, which are filled with tiny vesicles and are the specific points where information is transmitted.
Transmitted across the synapse.
Across the synapse, yes.
So neurons are the communicators. What about glial cells? They used to be thought of as just support staff, right?
Mm-hmm.
But our understanding has evolved.
Their role has definitely expanded dramatically in our understanding. While they do provide vital structural and metabolic support, we now recognize their crucial active roles. Glial cells regulate the content of the extracellular space, influencing neurotransmitters right at the synaptic junction, and even participating in neural signaling. They are active partners.
Active partners, okay.
One of their most critical jobs is forming the myelin sheath.
The insulation, like on an electrical wire, preventing signal loss.
Exactly. The myelin sheath is a phospholipid-based insulating layer that glial cells wrap along the length of the axon. Its primary purpose is to dramatically increase the speed and efficiency of electrical signal transmission. In the CNS, these myelin sheaths are formed by oligodendrocytes, while in the PNS, they are formed by Schwann cells. But this insulation isn't continuous, it's segmented.
Ah, the gaps, the nodes of Ranvier.
Precisely. Interspersed between the segments of myelin are tiny, exposed segments of the axon called nodes of Ranvier. What's crucial about these nodes is their high concentration of voltage-gated ion channels. These channels facilitate something remarkable called saltatory conduction.
Right.
Instead of the electrical signal or action potential having to propagate continuously down the entire axon slowly, it jumps rapidly from one node of Ranvier to the next, literally skipping the myelinated segments.
That saltatory conduction is truly mind-blowing, essentially creating a biological broadband network, dramatically speeding up signal transmission. It's like having dedicated express lanes for information, making our neural processing incredibly efficient. Beyond just structure, how do we functionally categorize the immense array of jobs the nervous system performs? It has such a vast array of responsibilities, from conscious action to automatic regulation.
Functionally, the nervous system is primarily organized into two broad realms: the somatic nervous system and the visceral nervous system. Think of them as handling different types of interactions, one for conscious, external engagement, and the other for unconscious, internal regulation.
Let's start with the somatic system. That sounds like conscious control and sensation.
You're right. The somatic nervous system consists of nerves that carry conscious sensory information from your peripheral regions, like touch on your skin, temperature, or the precise position of your limbs, back to the central nervous system. It also includes the motor nerves that exit the CNS to innervate your voluntary or skeletal muscles.
Okay.
So when you consciously decide to reach out and pick up a cup, that's your somatic nervous system executing that deliberate action. It's all about your conscious interactions with and awareness of the external world.
And the visceral nervous system is that its automatic, unconscious counterpart.
Exactly. The visceral nervous system, often synonymously called the autonomic nervous system, consists of nerves carrying sensory information into and motor commands out of the CNS to regulate your homeostatic functions. This is about the automatic unconscious control that keeps your internal environment stable and balanced.
Like heart rate, digestion.
Precisely. Heart rate, digestion, breathing, blood pressure, glandular secretions. You don't consciously control these vital processes. Your visceral nervous system expertly handles them automatically, ensuring your body maintains its delicate internal equilibrium. If we connect this to the bigger picture, these two systems highlight how the nervous system manages both our deliberate interactions with the world and the essential background processes keeping us alive. It's a constant, intricate dance of conscious control and automatic regulation.
All right, let's explore the crowning glory of our nervous system, the brain itself. We're starting with its outer shell, the cerebral hemispheres, often thought of as our thinking cap and the seat of consciousness.
Indeed. When we examine the brain's outer surface, the cerebral cortex, it's composed of 6 layers of neuronal cell bodies. This is what we refer to as gray matter, the primary site of neural computation. Deep inside the cerebral hemispheres, you find the myelinated axonal processes of these cells. Because myelin is a lipid-rich insulator, these large bundles appear whitish, hence they are called white matter.
So gray matter on the outside, white matter deeper inside. That's a key distinction compared to the spinal cord.
It is, and it's a crucial topographical difference. In the brain, gray matter forms the outer cortical surface, with white matter running deep, connecting these cortical areas. In the spinal cord, white matter is actually superficial to the gray matter. This reflects their distinct functional emphasis. Now, if you examine the surface of the cerebral hemispheres, you'll notice it isn't smooth. It's a complex landscape of elevations called gyri and infoldings called sulci.
Right, the folds.
These aren't just aesthetic, they significantly increase the surface area, allowing for vastly greater numbers of neurons and processing power within the limited confines of the skull.
It's like crumpling a piece of paper to fit more information onto it. And these aren't just one big amorphous mass, right?
Yeah.
They're clearly divided into distinct functional lobes.
That's right. Structurally, each cerebral hemisphere is divided into four major lobes. Anteriorly, we have the frontal lobe, separated from the parietal lobe by the central sulcus. Laterally, the frontal lobe is separated from the temporal lobe by the lateral sulcus, or sylvian fissure.
Okay.
The parietal lobe is posterior to the frontal lobe. The occipital lobe is at the very back. Medially, they're separated by the parieto-occipital sulcus. And the temporal lobe is inferior to both the frontal and parietal lobes. Along the midline, the two hemispheres are separated by the deep longitudinal fissure.
And there's a hidden part of the cortex as well, tucked away.
Yes, if you gently pry open that lateral sulcus, you'll reveal a small, distinct area of cortex called the insula. It's concealed laterally by parts of the frontal, parietal, and temporal lobes, collectively the operculum, meaning lid. The insula has unique connections involved in taste, visceral sensation, emotion, a whole range of functions. This intricate lobular organization, well, it raises an important question about how these distinct lobes, with their unique functional specializations, still manage to work together so seamlessly.
Now that we've established the geography of the brain, how does all this information get around within the brain itself and to other parts of the CNS? It's like a superhighway system, directing immense traffic between all those specialized regions.
That's an excellent analogy for the complex network of white matter pathways. Starting right beneath the cortical gray matter, you find a broad, fan-shaped expansion of white matter called the corona radiata. This is where many axons begin their journey.
So a broad fan of fibers that then converges.
Exactly. This broad pathway then condenses into a more compact, absolutely crucial structure called the internal capsule. When viewed in horizontal sections, it has a distinct V shape. This internal capsule is vital because it contains nearly all the axons traversing to and from cortical regions and deeper nuclear structures.
A critical hub for neural traffic.
Absolutely. A highly vulnerable choke point for major motor and sensory pathways. It's typically divided into three parts based on anatomy. The anterior limb is bounded by the caudate head medially and the lentiform nucleus laterally. This transitions into the genu, Latin for knee, around the level of the interventricular foramen. Then it completes its course as the posterior limb, lateral to the thalamus and medial to the lentiform nucleus. It's a dense bundle of communication lines.
And connecting the two cerebral hemispheres, we have another major white matter pathway.
That would be the corpus callosum. Unlike the more vertical pathways of the internal capsule, the corpus callosum is a massive horizontally running structure. It's formed by myelinated axons that specifically link the two cerebral hemispheres. It's the largest commissural pathway.
Allowing the two sides of the brain to talk.
Exactly, facilitating cross-hemispheric communication, coordinating tasks, and ensuring they work as a cohesive whole. It has distinct parts, too. The rostrum, genu, body, and splenium.
So the corpus callosum is like the ultimate bridge, allowing the two halves of our brain to talk to each other constantly. It's astounding how precisely these bundles are organized. Beyond the solid structures of gray and white matter, our brain also has these fascinating fluid-filled chambers, the ventricular system. What are they, and what crucial role do they play in brain health?
That's right, the ventricular system. This intricate network of caverns and canals is derived directly from the inner canal of the developing neural tube we discussed. As the brain grows and folds, these cavities adapt their shape to the surrounding structures.
So the very first hollow of the neural tube becomes this complex system.
Precisely. The most rostral are the two large C-shaped lateral ventricles deep within the cerebral hemispheres. They extend through all the major lobes and have 5 named parts: anterior horn, body, posterior horn, inferior horn, and the atrium where they meet.
That sounds like a lot of intricate plumbing. What about the fluid itself, and where is it produced?
Lining most of these ventricles is a specialized tissue called the choroid plexus. This series of modified ependymal cells actively produces cerebrospinal fluid, or CSF. In adults, it generates about half a liter of CSF every day.
Half a liter a day.
Yeah.
So how does this CSF circulate through this complex system, and why is that flow so important?
The flow is elegantly directed. From the 2 lateral ventricles, CSF flows through the interventricular foramen of Monro into the slit-like third ventricle surrounded by the thalamus and hypothalamus. The third ventricle then connects to the fourth ventricle via the cerebral aqueduct, which runs through the midbrain. The fourth ventricle is positioned between the pons medulla anteriorly and the cerebellum posteriorly. From here, CSF exits into the subarachnoid space through 3 openings: the 2 lateral foramina of Luschka and the single midline foramen of Megendi. If we connect this to the bigger picture, the constant flow of CSF is not just about cushioning, it's vital for nutrient delivery and waste removal, maintaining the brain's delicate environment. It provides hydraulic support, absorbs shock, and critically maintains the stable chemical environment, homeostasis, essential for brain function.
Think of our brain as the most precious cargo, requiring multiple layers of robust protection. That's where the meninges come in. These specialized coverings keep our brain safe within the skull.
That's an excellent analogy. The CNS is enveloped by three concentric connective tissue coverings called meninges. They provide support, stabilize the brain, and create a protective environment. We'll focus on the cranial meninge first.
So what's the outermost layer, the brain's first line of defense?
The outermost is the dura mater, Latin for hard mother. It's a remarkably tough, fibrous, double-layered sheet. The outer periosteal layer is fused to the inner surface of the skull. The inner meningeal layer lies against the arachnoid mater. In certain regions, these layers separate to form dural venous sinuses, crucial for draining venous blood.
And these layers can create spaces, right? Especially with trauma.
Yes, there are two potential spaces. The epidural space is superficial to the periosteal layer, between dura and skull. The subdural space is deep to the meningeal layer. Both can fill with blood during head trauma, causing serious conditions like epidural or subdural hematomas. What's also fascinating is that the meningeal dura folds in on itself to form dural reflections or septa.
Like internal bracing for the brain.
Exactly. These partitions divide the cranial cavity and restrict excessive brain movement. They include the falx cerebri between the cerebral hemispheres, the tentorium cerebelli separating cerebrum from cerebellum, the smaller falx cerebelli, and the diaphragma celli covering the pituitary fossa.
So an incredibly tough outer layer with internal partitions. What's next moving inward?
Deep to the dura is the arachnoid mater, named for its spiderweb appearance. Its outer layer sits adjacent to the dura. Delicate web-like strands, arachnoid trabeculae, extend from this outer layer internally to connect to the pia mater.
And the pia mater, the innermost and most delicate layer.
The pia mater, or tender mother, is indeed the innermost. It's an exquisitely thin, veil-like membrane that intimately follows every gyrus and sulcus, adhering closely to the brain surface.
Like cling wrap?
Just like cling wrap. Between the pia and arachnoid is the crucial subarachnoid space. This space contains the vital cerebrospinal fluid and houses the major blood vessels supplying the brain.
It's truly an incredibly tough, yet delicately cushioned system engineered to provide multi-layered protection for our most vital organ. Our brain is an incredibly high-demand organ consuming a disproportionate amount of the body's energy and oxygen, so it absolutely needs a robust, redundant delivery system. Let's talk about the brain's lifeline, its cerebral vasculature.
You're right, the brain cannot tolerate even brief interruptions in blood supply. Its supply is divided into two main systems: the anterior circulation from the internal carotid arteries and the posterior circulation from the vertebral arteries. The internal carotid arteries arise from the common carotids in the neck, course through the neck, carotid canal, petrous temporal bone, cavernous sinus, and finally enter the subarachnoid space near the optic chiasm.
That's a long, winding road. What are its major branches once it reaches the brain?
It gives rise to the ophthalmic artery, posterior communicating artery, and anterior choroidal arteries. Its two major terminal branches are the anterior cerebral artery, ACA, and the middle cerebral artery, MCA.
So that's the anterior half. How does the posterior circulation contribute?
The posterior cortex, brainstem, and cerebellum get supply from the vertebral basilar system. Two vertebral arteries arise from the subclavian arteries, ascend through the cervical vertebrae. At the pontomedullary junction, they unite to form the single basilar artery, which runs along the ventral brainstem.
And the basilar artery's main terminal branch.
At the midbrain level, the basilar artery terminates as the two posterior cerebral arteries, or PCAs. These PCAs turn posteriorly, supplying the inferior and medial temporal and occipital lobes. Crucially, the posterior communicating artery connects the PCA to the internal carotid. This connection, along with the anterior communicating artery linking the two ACAs, forms a critical anastomotic ring.
The circle of Willis, that's the famous one, providing redundancy, right?
Exactly. The circle of Willis is this critical ring providing collateral circulation. If one major feeder vessel is compromised, blood can still reach vital areas through alternative routes. It's an incredible built-in redundancy system.
Incredible. And what are the specific territories these major arteries supply?
Let's break those down quickly. The anterior cerebral artery, ACA, courses medially in the longitudinal fissure, supplying most of the medial aspect from frontal to anterior parietal lobe. The middle cerebral artery, MCA, branches laterally into the lateral fissure, gives off lenticulostriate arteries to deep structures, then bifurcates. Its superior division supplies the lateral, frontal, and anterior parietal lobes. The inferior division supplies the temporal and anterolateral parietal lobes. MCA covers much of the lateral surface.
On the PCA.
And the posterior cerebral artery, PCA, perfuses the inferior and medial temporal and occipital lobes. This intricate network, especially a circle of Willis, is a stunning example of biological redundancy. It helps ensure continuous blood supply, which is why understanding it is crucial for dealing with strokes.
Just as important as getting blood in is getting it out. And the brain has an equally complex system for venous drainage.
Absolutely. Venous drainage occurs through superficial veins, deep veins, and dural venous sinuses, ultimately leading to the internal jugular veins. A key distinction is that cerebral veins and dural sinuses lack valves.
No valves. That's quite different. So how do they drain efficiently without them?
The veins connect to the dural venous sinuses, which are channels between dural layers. The superior sagittal sinus runs along the top of the falx cerebri, collecting blood from superior veins, draining into the transverse sinuses. Each transverse sinus turns inferiorly to form the sigmoid sinus, which exits the skull to become the internal jugular vein.
And the deeper veins.
Along the inferior margin of the falx is the inferior sagittal sinus. Posteriorly, it joins the great cerebral vein of Galen, which collects deep blood, to form the straight sinus. The point where the straight superior sagittal and occipital sinuses converge is the confluence of the sinuses, usually drained by the transverse sinuses.
So a large system of sinuses acting as collecting points.
Exactly. While superficial vein patterns vary, three are fairly constant. The superficial middle cerebral vein, temporal lobe to cavernous sinus. The superior anastomotic vein of Trollard connects middle cerebral to superior sagittal, and the inferior anastomotic vein of Labbé connects middle cerebral to transverse sinus. These provide alternative drainage routes.
Okay.
In contrast, the deep veins are more consistent. Most drain into the great cerebral vein of Galen. For instance, the anterior cerebral and deep middle cerebral veins join to form the basal vein of Rosenthal. Septal and thalamostriate veins join to form the internal cerebral veins. Both the internal cerebral veins and basal veins converge posterior to the midbrain to form the great vein of Galen.
And we also need to mention the cavernous sinus again, clinically important.
Yes, the cavernous sinus is a unique plexus of veins on either side of the pituitary fossa. It receives ophthalmic veins from the eye and drains into the superior and inferior petrosal sinuses. Its location makes it vulnerable to infection from the face and cranial nerve compression.
So an elegant valveless plumbing system ensuring constant flow, quite different from the rest of the body, highlights the brain's specialized needs. Now, let's move to the brainstem, often called the reptilian brain for its vital primal functions. This is the critical bridge connecting the higher processing centers to the spinal cord. This is where the absolute essentials happen.
That's the perfect way to frame it. The brainstem is a stalk-like structure in the posterior cranial fossa, linking the forebrain rostrally with the spinal cord caudally. From rostral to caudal, it's composed of three segments: the midbrain, the pons, and the medulla oblongata.
And its functions are truly foundational then.
Broadly, yes, three main indispensable functions. First, it's a crucial conduit for ascending sensory tracts and descending motor tracts. All traffic passes through here.
Right.
Second, it houses nuclei for most cranial nerves 3 through 12, though CNK has components in the cervical spinal cord. And third, the brainstem contains vital reflex centers regulating respiration, cardiovascular function, and consciousness.
Let's unpack this with a closer look at each segment, from the outside in. What are the key external features? Hashtag to one. External features of the brainstem.
Starting with the midbrain, it's about 2 centimeters long, connecting forebrain to pons and cerebellum. Anteriorly, there's the deep interpeduncular fossa flanked by the crus cerebri, descending motor fibers. Cranial nerve the third, oculomotor, emerges medially from the crus. Posteriorly, the prominent superior and inferior colliculi 4 bumps for visual auditory reflexes. Cranial nerve the 4, trochlear, emerges between the inferior colliculi, crosses, and wraps laterally.
Moving down to the pons, what are its distinguishing features?
Caudal to the midbrain, the pons is about 2.5 centimeters long, connecting midbrain to medulla and bridging to the cerebellum. Anteriorly, the shallow basilar groove for the basilar artery. Its prominent convex shape is due to pontocerebellar fibers bridging to the cerebellum via the middle cerebellar peduncle.
Very distinct.
Anterolaterally, CNV, the large trigeminal nerve, emerges. At the pontomedullary junction, from medial to lateral, CNs viciscens, abducent, 7, facial, and 8 vestibular cochlear emerge.
And finally, the medulla oblongata, transitioning into the spinal cord.
The medulla is the longest, about 3 centimeters. Broad rostrally, tapers caudally to the spinal cord at the foramen magnum, anteriorly the anterior median fissure. Lateral to it are the medullary pyramids, descending motor fibers. Caudally, these give way to the decussation of the pyramids where most motor fibers cross.
The crossing point.
Lateral to the pyramids are the olives, underlying inferior olivary nuclei. Rootlets of CN12, hypoglossal, emerge at the pyramidal junction. Posteriorly, it forms the floor of the fourth ventricle, showing gracile and cuneate tubercles, sensory relays. Rootlets of CNIX, glossopharyngeal, X, vagus, and XI spinal accessory emerge near the olive inferior cerebellar peduncle junction. This external topography is a rich roadmap. Just observing these features tells us so much about major pathways and cranial nerve functions. Hashtag 2, internal architecture of the brainstem.
That's a comprehensive external view. Now let's peel back the layers and see what's happening inside, like looking at the engine room.
Exactly. Internally, the brainstem can be divided into the tectum, roof, tegmentum covering, and basis, anterior portion. We rely on stained serial sections, often for myelin, to see the nuclei in tracks.
Let's start with the midbrain's internal structure.
The tectum includes the colliculi and cerebral aqueduct. A rostral section, superior colliculus level, shows the superior colliculi, aqueduct, periaqueductal gray, and nuclei like oculomotor, Edinger-Westphal, red nucleus, mesencephalic CNV, and substantia nigra. Key fibers include ascending lemnisci and descending crus cerebri. A caudal section, inferior colliculus level, shows the inferior colliculus, trochlear nucleus, and the decussation of the superior cerebellar peduncle.
So many critical structures packed in. Moving into the pons?
In the rostral pons, you see pontine nuclei, transverse pontocerebellar fibers, and longitudinal corticospinal fibers. The medial lemniscus is prominent. You also find the spinothalamic tract, MLF, and locus coeruleus. Mid-pons level shows the superior and middle cerebellar peduncles, CNV nuclei, and the trapezoid body, auditory. Caudal pons reveals the abducent nucleus, medial vestibular nucleus, and cochlear nuclei.
And finally, the medulla oblongata internally.
It's complex. Rostral medulla, inferior olivary level, has nuclei for CNs 8 to 12, the medial lemniscus, and the large inferior cerebellar peduncle. Vital respiratory cardiovascular centers are here.
Crucial area.
Moving caudally, internal arcuate fiber level, the gracile and cuneate nuclei are prominent. Their axons decussate as internal arcuate fibers, forming the medial lemniscus. Finally, at the caudal medulla, you see the pyramidal decussation where motor fibers cross before the spinal cord transition. This raises an important question. How can such small, dense areas have so many critical nuclei and pathways, each with a specialized role? It's a marvel of packing efficiency. Hashtag 3. Vascular supply to the brainstem.
Just like the rest of the brainstem demands a robust and dedicated blood supply, ensuring these vital functions are never interrupted.
That's right. Supply comes entirely from the vertebrovascular system. 2 vertebral arteries merge to form the basilar artery.
So the vertebral arteries are the foundation. What branches do they give off before merging?
Before merging, each gives off the posterior inferior cerebellar artery, PICA, crucial for the lateral medulla and inferior cerebellum. They also contribute to the anterior spinal artery and give off posterior spinal arteries. Medial anterior medulla gets supply from vertebral and anterior spinal branches.
And as the basilar artery ascends, what are its key branches?
At the caudal pons, the anterior inferior cerebellar artery, AICA, branches off, perfusing lateral caudal pons and anterior lateral cerebellum. Rostral lateral pons get supply from circumferential branches, medial pons from paramedian branches. Just before the midbrain, the superior cerebellar arteries branch off, supplying superior cerebellar peduncles, caudal dorsal midbrain, and superior cerebellum. Finally, the terminal branches are the posterior cerebral arteries, PCAs, perfusing lateral midbrain, thalamus, and medial occipital inferior temporal lobes.
It's a marvel of engineering, truly, how this system ensures highly localized blood flow to even the deepest parts. Our nervous system doesn't end at the brainstem, continues as this incredible conduit, the spinal cord, the body's primary information superhighway.
That's a perfect description. The spinal cord is continuous with the medulla, starting near the foramen magnum. It's cylindrical within the vertebral canal, extending down to about L1, L2 vertebrae in adults. Its primary function is as a conduit for ascending sensory and descending motor tracts, connecting brain and body for movement, reflexes, and sensation.
Let's unpack this crucial connection, starting with its protective layers, the spinal meninges.
Just like the brain, the spinal cord has three meninges: dura, arachnoid, and pia mater. The spinal dura is continuous with the cranial dura's inner layer, extending down to S2.
Way beyond the cord itself.
Exactly. A unique feature is the epidural space separating spinal dura from the vertebral canal, containing fat and veins. The subdural space is potential. The arachnoid is similar but less adherent. The innermost pia mater is highly vascular, adhering closely to the cord surface. Midway between nerve roots, the pia forms the denticulate ligament, anchoring the cord centrally within the subarachnoid space.
Preventing it from moving too much.
Right. And the subarachnoid space itself, containing CSF, also extends down to S2. Since the core ends at L1, L2, this creates an enlarged, safe space below the cord for clinical CSF access, like lumbar punctures. This safe space is a critical anatomical detail for clinical procedures, highlighting the elegance of nature's design. #2. External features of the spinal cord. Externally, the cord has longitudinal fissures and sulci. The anterior median fissure is deep. The posterior median sulcus is shallower, flanked by posterolateral sulci.
And from these surfaces, the nerves emerge.
Yes. Rootlets emerge, coalesce into anterior motor and posterior sensory roots, which converge to form 31 pairs of spinal nerves along its length. The cord isn't uniformly thick. Two regions are enlarged for limb innervation.
The enlargements for our arms and legs.
Precisely. The cervical enlargement, C5T1, serves the upper extremities. The lumbar enlargement, L2S3, serves the lower extremities. These reflect the increased circuitry needed.
It's a beautiful functional symmetry, adapting its size to meet complex demands. #tag3. Internal features of the spinal cord.
In cross-section, the spinal cord shows a distinct pattern. Inner H-shaped gray matter, neuronal cell bodies, surrounded by outer white matter, myelinated axons.
So the opposite of the brain's cortex. And
Yeah.
the gray matter has distinct horns.
Yes. Ventral anterior horns contain motor neuron cell bodies. Dorsal posterior horns receive sensory information. There's also the intermediolateral cell column, T1L2 region, housing preganglionic sympathetic neuron cell bodies. The gray matter is further divided into 10 zones, Wechs's laminae, reflecting its diverse cytoarchitecture.
And the white matter surrounds this, carrying signals up and down.
Indeed. The white matter is divided into funiculi. The anterior funiculus has motor axons. The posterior funiculus has sensory axons. The lateral funiculus is mixed. Each funiculus houses specific tracts for distinct information transfer. This raises an important question about how this intricate internal organization allows for such precise and rapid communication throughout the body. It's highly organized.
Let's follow the information superhighway in reverse, tracing how sensations travel from fingertips and toes all the way up to conscious perception.
Right. Sensory information enters the CNS via a series of neurons, synapsing in the cord, cortex, and elsewhere. Modalities include pain, temperature, touch, proprioception.
And how do we differentiate conscious versus subconscious input?
Conscious perception travels specific pathways to the primary somatosensory cortex. Subconscious input goes to places like the cerebellum for coordination. For conscious perception, two major pathways ascend the spinal cord, each using 3 neurons.
What are those two major pathways?
First, the anterolateral pathways, conveying pain, temperature, and crude touch. Second, the posterior column medial lemniscal pathway for discriminative touch, vibration, and conscious proprioception. Let's start with the anterolateral.
Okay, the anterolateral pathway, pain, temperature, crude touch, basic sensations.
This pathway includes the spinothalamic, spinoreticular, and spinomesencephalic tracts. We'll trace the spinothalamic for conscious perception. The first-order neuron cell body is in a spinal ganglion. Its axon enters the posterior horn. Some synapse immediately on second-order neurons, laminae I and V. Others descend 1 to 2 segments in Lissauer's tract before synapsing.
So the signal comes in, synapses, and then crosses over in the spinal cord.
Exactly, and this crossing is critical clinically. The second-order axons cross obliquely over 2 or 3 segments in the anterior commissure to join the contralateral enterolateral tract. They ascend through the CNS to the third-order neuron cell bodies in the ventral posterior lateral nucleus, VPL, of the thalamus. From there, third-order axons project through the internal capsule's posterior limb to the primary somatosensory cortex for conscious perception.
And the spinoreticular and spinomesencephalic tracts, how do they contribute to pain experience?
They share a similar start. The spinoreticular tract projects to the reticular formation, conveying emotional arousal aspects of pain. The spinomesencephalic tract projects to the periaqueductal gray and superior colliculi, involved in central pain modulation, allowing the brain to suppress pain signals.
Fascinating how different aspects of pain sensation emotion modulation are handled distinctly. Now, the posterior column medial lemniscal pathway for fine touch and proprioception.
This is also a three-neuron chain, but with different routing. First-order neuron cell bodies are in a spinal ganglion. Their axons enter the posterior root and ascend ipsilaterally in the posterior funiculus, without crossing in the cord. Lower limb trunk info travels in the gressile fasciculus. Upper limb neck info in the cuneate fasciculus. They ascend all the way to the caudal medulla.
So no crossing in the spinal cord, unlike the spinothalamic.
Correct. They synapse with second-order neurons in the nucleus gracilis and cuneatus in the caudal medulla. Here, the second-order axons cross over as internal arcuate fibers. They form the contralateral medial lemniscus, ascending through the brainstem to the third-order neurons in the VPL of the thalamus. Finally, third-order axons project through the posterior limb of the internal capsule to the primary somatosensory cortex for conscious perception of fine details.
It's amazing how two distinct pathways, one for crude sensations and one for fine, detailed information, ascend simultaneously, each with precise routing. Allows incredible sensory sophistication. Now let's reverse the flow again. How does our brain tell our body what to do? The descending tracts are the master controllers of movement.
Indeed. Descending tracts handle voluntary movements, postural adjustments, coordination of head-neck eyes. They originate from cortex or brainstem, influenced by cerebellum basal nuclei feedback. We think in terms of upper motor neurons, UMNs, in the brainstem, and lower motor neurons, LMNs, in the spinal cord gray matter. UMNs command LMNs, which innervate muscles. We'll start with the lateral motor system for limb control.
So, the lateral motor system, what's included and what's its primary function?
It includes the lateral corticospinal tract and rubrospinal tract. Both are in the lateral column, synapsing on LMNs in the lateral anterior horn. They control voluntary, skilled movements of distal limbs.
Which one is most clinically important?
The lateral corticospinal tract, undoubtedly. It controls precise, voluntary movements of upper-lower extremities. UMN cell bodies are in the primary motor cortex. Axons converge in corona radiata, descend through the internal capsule's posterior limb, crus cerebri anterior pons, form the pyramid in the medulla.
And this is where the famous decussation happens.
Precisely. At the caudal medulla, most axons, 85 to 90%, decussate to the contralateral side, forming the lateral corticospinal tract. The rest form the anterior corticospinal tract, part of the medial system. After crossing, axons descend the cord, synapse on LMNs in the lateral anterior horn. LMN axons exit via anterior root to muscles. This crossing means left cortex controls right body, vice versa.
And the rubrospinal tract, how does it contribute?
UMNs start in the red nucleus, midbrain. Axons cross midline, ventral tegmental decussation, descend as rubrospinal tract through brainstem and lateral column to cervical regions. They synapse with interneurons to facilitate flexors and inhibit extensors, mainly in the upper limb. Assists in fine motor control.
Now for the medial motor systems. For broader movements, posture, and balance.
They regulate axial truncal muscles for posture, balance, gait, head-neck orientation. They primarily project bilaterally on interneurons. 4 key tracts here.
Starting with the anterior corticospinal tract, the uncrossed portion.
Correct. Formed by UMNs that didn't decussate. Descend ipsilaterally through medial anterior cord to upper thoracic, project bilaterally to interneurons in medial anterior horn, controls axial proximal limbs for posture.
And the tectospinal tract, involved with visual reflexes.
Tectospinal axons arise from the superior colliculus, dorsal midbrain. Axons decussate, dorsal tegmental decussation, form tectospinal tract along midline brainstem into cervical cord. Project bilaterally to interneurons, believed to modulate reflex postural movements to visual stimuli, like orienting to a flash.
What about the vestibulospinal tracts? Crucial for balance.
Absolutely right. Axons originate from vestibular nuclei pons medulla, receiving inner ear cerebellum input. Medial vestibulospinal tract from medial nucleus projects bilaterally to thoracic cord, head-neck positioning. Lateral vestibulospinal tract from lateral nucleus descends ipsilaterally entire cord, synapses on interneurons, facilitates extensor center-gravity muscles, inhibits flexors, crucial for balance posture.
And finally, the reticulospinal tracts, more general integrating role.
Axons arise from reticular formation, pons medulla. Descend ipsilaterally, anterior white matter. Synapse with interneurons. Regulate voluntary movements in reflex activity, influence autonomic outflow. Provide background tone, integrate motor commands with posture. This level of detail, comparing lateral and medial systems, highlights the brain's incredible specificity in controlling everything from typing to maintaining balance. Hashtag 6, vascular supply to the spinal cord.
Finally, let's complete our look at the spinal cord with its dedicated blood supply. Must be well perfused.
It is, but with a unique architecture. Supply is from 3 longitudinal vessels and segmental branches. The longitudinal are the single anterior spinal artery and 2 posterior spinal arteries.
Where do these originate and run?
2 posterior spinal arteries usually branch from vertebral artery or piscae, descend along posterolateral sulcus, supplying posterior third of cord. The single anterior spinal artery originates from union of 2 vertebral artery branches in cranial cavity, descends along anterior median fissure, supplying anterior 2/3.
So these main arteries run the length. Are there reinforcing vessels?
Yes, crucial reinforcement comes from 8-10 segmental medullary arteries entering at various levels. One is particularly important, the largest, the artery of Adamkiewicz, usually on the left, lower thoracic upper lumbar region, significantly perfuses lower spinal cord. Damage here can be devastating.
The artery of Adamkiewicz is a particularly important clinical detail, a single critical vessel, highlighting a potential vulnerability.
Venous drainage is via longitudinal channels connecting with anterior-posterior spinal veins on the cord surface, eventually draining to larger plexuses. Like cerebral veins, they are generally valveless.
Moving deeper into the brain, we encounter a crucial structure often called the gateway to the cortex, the thalamus. What does this inner chamber do?
The thalamus is a large egg-shaped gray matter mass from the diencephalon. Its most significant role is as a synaptic relay station for virtually all sensory and motor pathways projecting to the cortex. Almost everything goes through here first.
So all signals pass through on their way to conscious awareness?
Almost all. But importantly, it's not just passive relay. It actively functions as a gatekeeper or filter, preventing or enhancing info transfer based on behavioral state. Think filtering background noise. Sensory, motor, limbic, modulatory signals all relay here. It's highly dynamic.
A powerful filter. What about its location? Very central.
It is indeed. Anteriorly, contacts interventricular foramen, forms lateral walls of third ventricle with hypothalamus. Lateral is the posterior limb of internal capsule. Dorsal is body of lateral ventricle, caudally connects to midbrain, 2 masses interconnected by interthalamic adhesion.
So a true central hub, functionally and anatomically.
To understand it better, its nuclei are classified structurally and functionally. Structurally, based on relation to the Y-shaped internal medullary lamina, anterior, medial, lateral, intralaminar groups.
And functionally, how do they perform their duties?
Functionally, 3 classes: relay, intralaminar, reticular. Most are relay nuclei with reciprocal excitatory connections to cortex. Relay nuclei are subdivided into specific, project to distinct primary sensory motor cortex, and non-specific diffuse projections. Specific relay nuclei are mostly in lateral thalamus. All sensory modalities except olfaction relay here.
Can you give examples of those specific relays?
Certainly. Ventral posterior lateral, VPL nucleus, receives body limb somatic input. Ventral posterior medial, VPM, gets head somatic input. Both project to somatosensory cortex. Medial geniculate body relays auditory info. Lateral geniculate body relays visual info. Association nuclei like dorsomedial DM connect to prefrontal cortex for olfactory limbic functions.
And the other classes, intralaminar and reticular.
Intralaminar nuclei are within the internal medullary lamina. Reciprocal projections mainly with basal nuclei and reticular formation. Rostral ones influence arousal via ascending reticular activating system. Caudal ones, including centromedian nucleus, involved in basal ganglia circuitry for movement regulation.
And the reticular nucleus, quite distinct.
Yes, the reticular nucleus is a thin sheet along the lateral thalamus. Critically, it does not project to cortex. It receives input from other thalamic nuclei and cortex, then projects back to the thalamus. Its GABAergic neurons regulate thalamic activity.
A feedback loop.
Exactly. This unique feedback-only loop allows it to be a powerful regulator, fine-tuning what information gets processed, key for attention. This raises an important question. How does the reticular nucleus's unique feedback-only loop allow it to be such a powerful regulator of thalamic activity, essentially fine-tuning what information is processed? Hashtag 2. Vascular supply to the thalamus. Given its critical role, the thalamus gets diverse arterial supply from penetrating branches from ACA, anterior choroidal artery, internal carotid, lenticulostriate arteries, MCA, and thalamoperforator arteries, PCA. This multi-source supply underscores its importance.
So a critical hub like the thalamus has a robust, multi-source blood supply, underscoring its importance. Deep within the brain, another fascinating collection vital for smooth movement, posture, even motivation, the basal nuclei.
That's right. Gray matter structures deep in the forebrain base. They play a significant role in controlling posture and voluntary movement via connections to thalamus, cortex, other basal nuclei. They also connect to the limbic system, influencing behavior and motivation.
So involved in mechanics and the why behind movement.
Exactly. Studying them requires horizontal and coronal sections to appreciate their 3D shapes and relationships. Their deep location makes them central.
So what does this striped body actually consist of? Hashtag one. Key structures of the basal nuclei.
The term corpus striatum, or striped body, includes the caudate nucleus and the lentiform nucleus, named for the striated bands interconnecting caudate and putamen through the internal capsule's anterior limb.
And the lentiform nucleus.
Lentiform means lens-shaped. It includes the globus pallidus and putamen, located lateral to the internal capsule, which separates them from thalamus and caudate medially, laterally bordered by external capsule, claustrum, and insula.
And the caudate nucleus, with its C-shape.
Yes, the caudate is medial to the internal capsule, a large C-shaped structure following the lateral ventricle's curve. Divided into head, body, tail. Head is large, rounded, forms lateral wall of anterior horn, continuous with putamen rostrally. Because of this close relationship, putamen and caudate are often called the striatum key input area.
And the body and tail.
Head transitions to body near interventricular foramen. Body narrows, forms floor of lateral ventricle. Transitions to tail near posterior thalamus. Tail continues anteriorly in roof of inferior horn, terminating in the amygdaloid nucleus, linking to emotion. This structural complexity highlights its integral role within the deeper brain architecture. The basal nuclei operate through finely tuned input-output. Input is primarily to the striatum, catetaputamine, from cortex, thalamus, subthalamic nucleus, brainstem, substantia nigra. Output predominantly leaves from globus pallidus. Two key loops explain their influence, the direct and indirect pathways.
So like an accelerator and a brake for movement.
A very good way to think about it. The direct pathway results in an increase in motor activity, facilitates desired movements. Input to striatum, connections to globus pallidus, output to thalamus, back to cortex, allows smooth execution.
And the indirect pathway, the opposite.
Precisely. Similar course, but adds output connections to the subthalamic nucleus before globus pallidus. This results in an overall decrease in motor activity, acts like a brake, suppressing unwanted movements, refining motor programs.
And dopamine is critical for balancing these pathways.
Absolutely. Dopamine from the substantia nigra modulates both. Excitatory to D1 receptors, direct pathway, increases movement. Inhibitory to D2 receptors, indirect pathway, reduces braking. This complex interplay allows smooth, coordinated movement. Dopamine dysfunction, like in Parkinson's, causes motor symptoms, starkly showing its importance.
So these two pathways are like accelerator and brake pedals, constantly balancing, with dopamine as a crucial fine-tuning modulator. Nestled at the back, almost a little brain of its own, is the cerebellum, master of coordination, precision, and balance.
That's apt. Largest hindbrain structure. Two hemispheres connected by the vermis, midline. In posterior cranial fossa, covered by tentorium cerebelli, plays crucial role maintaining balance, influencing posture, coordinating movements by synchronizing muscle contraction and relaxation. The ultimate choreographer.
So refining movement, not initiating it.
Precisely. An error detector compares intended versus actual movements via feedback, sends corrective signals for smoothness and accuracy. Let's look at structure first. This raises an important question about how this intricate structure achieves such precise timing and coordination, essential for everything from walking to playing music. ###1. Structural organization of the cerebellum. It connects to the posterior brainstem via superior, middle, inferior cerebellar peduncles, input-output. Forms a roof of fourth ventricle. Surface has folds, folia, separated by fissures. Two key fissures divide it into three lobes. Superiorly, primary fissure separates anterior from posterior lobe. Inferiorly, posterior lateral fissure defines flocculonodular lobe, flocculus plus nodule, crucial for balance eye movements. Horizontal fissure borders superior inferior surfaces.
And internally.
Each folia has central white matter core covered by thin gray matter cortex. Branching pattern is the arbor vitae, tree of life. Deep within white matter are four pairs of nuclei output centers. Dentate, emboliform, globose, vestigial, lateral to medial, emboliform plus globose, interposed nuclei.
So deep nuclei are the output centers.
Correct. Output predominantly from these nuclei, leaving via superior cerebellar peduncle. Key principle. Each hemisphere coordinates movement on the ipsilateral side. Functionally, cortex has three areas. Vermis, midline, influences axial movements, neck, trunk. Intermediate zone controls distal limb muscles. Lateral zone aids motor planning for sequential movements, cognition.
How does the cortex process information?
Unique cellular architecture is key. Input comes via middle inferior peduncles as mossy fibers or climbing fibers from inferior olive. Mossy fibers excite granule cells. Granule cell axons become parallel fibers in molecular layer, synapsing on Purkinje cells.
And the climbing fibers.
Climbing fibers project directly, forming powerful excitatory connections with Purkinje cells. Purkinje cells are the main output neurons of the cortex, projecting inhibitory signals to the deep cerebellar nuclei. This is the final cortical processing stage.
It's like an incredibly complex feedback loop, constantly taking in info, processing, sending out precise adjustments for movement. Input pathways to the cerebellum. So where does the cerebellum get all this vast information? What are the key input pathways?
Three major sources: cerebral cortex, spinal cord, brainstem. Input from cerebral cortex relates to voluntary muscle control, movement coordination. Axons descend via internal capsule, terminate on pontine nuclei. Pontine axons cross over, enter contralateral cerebellum via middle cerebellar peduncle.
And from the spinal cord, critical feedback.
Input from spinal cord conveys proprioceptive info from muscles joints, for muscle tone, posture, body position awareness. Primary pathways: anterior and posterior spinocerebellar tracts. Originate from mechanoreceptors, ascend spinal cord, enter ipsilateral cerebellum via inferior cerebellar peduncle. Provides real-time updates. And brainstem input for balance.
Yes, from vestibular nuclei and reticular formation. Involved in reflexive balance, posture, gaze stabilization. Projections to ipsilateral cerebellum via inferior cerebellar peduncle. Feeds info on head position movement, arousal state.
This constant stream of input paints a picture of the cerebellum as a tireless integrator. Tag 3. Efferent output pathways from the cerebellum. So input's covered. How does it send refined commands back out? What are the efferent pathways?
Output originates from the four deep nuclei: dentate, ambuliform, globose, vestigial. They influence motor system to correct errors. Largest output from dentate nucleus, planning, initiating voluntary movement. Axons project to contralateral lateral dianal nucleus of thalamus after decussating a superior peduncle. Thalamus projects to motor cortex, influences posture, skilled movement.
And the interposed nuclei.
Ambuliform and globose, interposed, have similar course but additional target. Axons decussate in superior peduncle synapsed on contralateral VO thalamus, A&D contralateral red nucleus, midbrain. Red nucleus descends to inferior olive, monitors, corrects ongoing limb motor activity. And the vestigial nucleus.
Vestigial has broader projections: vestibular nuclei, reticular formation, contralateral VL thalamus, contralateral tectum, via inferior peduncle to ipsilateral vestibular nucleus, contralateral vestibular nucleus via uncinate fasciculus, reticular formation, via superior peduncles to contralateral tectum and contralateral VL thalamus. Influences balance, posture, eye movements. The fact that cerebellum's output crosses twice only to influence the ipsilateral side is fascinating. A sophisticated double cross ensuring precise control. Hashtag 4. Vascular supply to the cerebellum.
Another critical area, another robust blood supply. How is the cerebellum perfused?
By the same vertebro-basilar system supplying the brainstem. Vertebral arteries give off posterior inferior cerebellar artery, PICA, perfusing inferior cerebellum and lateral medulla.
And for the basilar artery?
Basilar gives off anterior inferior cerebellar artery, AICA, at caudal pons, perfusing anterior lateral cerebellum, middle inferior peduncles. Just before midbrain, superior cerebellar arteries branch off basilar, supplying superior peduncles and superior cerebellum. Comprehensive supply.
Another critical area, another highly specified and robust blood supply. Now let's turn to one of our most powerful senses, vision. How do we transform light into the vibrant world we perceive? Far more complex than just a camera.
The visual system is truly complex. Journey begins in the eyeball, then connections to thalamus, brainstem, primary visual cortex, association cortices. Mediates basic perception and higher functions like spatial relationships, face recognition. We'll focus on the primary geniculate pathway for conscious sight. What's fascinating here is the journey involves precise transformations at every step. Information is processed, inverted, split, recombined. Anterior structures prepare and focus light. Cornea, transparent front layer, does most initial refraction. Behind it, aqueous humor fills anterior chamber, between cornea pupil.
And the pupil acts like a camera's aperture.
Exactly. Controls light entry. Light then passes through the lens, clear by convex. With ciliary body separates anterior-posterior eye. Lens changes shape via ciliary muscle accommodation, parasympathetically controlled, to focus on different distances.
So a self-adjusting optical system, what's behind the lens?
Light traverses vitreous humor, jelly-filling posterior chamber, projected onto retina layers at the back. Retina has non-neural pigment layer and neural cell layers. Choroid layer lines sclera, and pigment epithelium absorb excess light.
And the actual light detectors?
Photoreceptors, rods and cones, interdigitate with pigment cells, transduce light into electrical signal, phototransduction. Importantly, images on retina are inverted vertically and laterally. Visual field defined by 4 quadrants.
Tell us more about rods and cones.
Rods, very sensitive for dim light vision, black-white peripheral. Outnumber cones, 20.1, concentrated in periphery. Cones for color vision, high acuity, fine detail. Predominate near macula. Fovea, maximal acuity, has only cones. Photoreceptors contact bipolar cells, first-order neurons. Bipolar cells synapse with ganglion cells, second-order neurons.
Other cells modulating this early processing.
Yes, horizontal and amacrine cells. Transverse interneurons modulate bipolar ganglion cell excitation, refining signal, enhancing contrast before it leaves the eye.
It's a miniature camera, self-adjusting, incredibly sensitive, doing complex processing before the signal even leaves the eye.
After retinal processing, the journey continues. Ganglion cell axons coalesce at the optic disc. No photoreceptors, it is a blind spot. Axons leave optic disc, form optic nerve, acquires myelin, oligodendrocytes, and meninges, defining it as CNS component.
So an extension of brain tissue. Then they meet at the optic chiasm.
Correct. Optic nerves converge anterior to infundibular stalk at optic chiasm. Critical decussation occurs here. Axons from nasal retina, peripheral visual field, cross to contralateral optic tract. Axons from temporal retina, central visual field, stay ipsilateral. Fundamental for perception.
So each side of the brain now receives info from the opposite visual field.
Precisely. Optic tracts course posteriorly around midbrain. Vast majority enter lateral geniculate nucleus, LGN, of thalamus for a synaptic relay. Primary relay for conscious vision. Small portion bypasses LGN to protectal area superior colliculus for pupillary light reflex and coordinating eye movements.
And from the LGN.
Axons leave LGN, form optic radiations, continue to primary visual cortex and occipital lobe. Lower optic radiation fibers, lower visual cortex. Upper fibers, upper cortex.
So how does the inversion and crossing translate to our unified perception?
Due to retinal inversion and chiasm decussation, optic tracts, thalamus radiations, primary visual cortex receive info only from the contralateral half of the visual field. Right cortex processes left field, vice versa. Understanding this pathway is essential clinically for identifying lesions based on visual field deficits, patterns mapped to specific damage locations. This raises an important question. How does the brain then reinvert and combine the inverted, split, and rerouted information from both eyes to create a seamless, unified visual experience?
Let's explore 2 more incredible sensory systems, hearing and balance, both originating in the complex inner ear.
Indeed. Cranial nerve 8th, vestibulocochlear, conveys info from vestibular balance and auditory organs to pontomedullary junction. While traveling together, these sensory functions have distinct central pathways once they reach the brainstem. What's fascinating here is the sheer mechanical precision involved in translating air vibrations and head movements into electrical signals the brain interprets. Sound journey. Pinna directs waves to external acoustic meninges ear canal, tympanic membrane, eardrum. Membrane vibrations transmitted to middle ear ossicles, malleus, innicus, stapes, amplifying sound. Tensor tympanis stapedius muscles dampen loud sounds.
Mechanical amplifier protector, then into the cochlea for electrical conversion.
Exactly. In cochlea, waves converted at organ of Corti. Rests on basilar membrane scala media, endolymph. Basilar membrane separates scala vestibuliscula tympani paralymph. Sound waves move paralymph, displace basilar membrane, deflect hair cells, sensory receptors. Hair cells synapse with sensory neurons, cell bodies and spiral ganglion and modiolus. Axons form cochlear nerve, internal acoustic meatus, pontomedullary junction.
Mechanical vibration to electrical signal. What happens in the brainstem?
Central pathways. First-order axons terminate ipsilaterally on dorsal ventral cochlear nuclei. Second-order ascending axons go to pons, project bilaterally to superior olivary nucleus, important for sound localization. Fibers continue as lateral lemniscus, inferior colliculus, midbrain relay. Axons from inferior colliculus relay in medial geniculate nucleus, thalamus. Final target, primary auditory cortex, superior temporal gyrus, tonotopically represented throughout pathway.
Hearing pathway, but there's a feedback loop too.
Yes, crucial descending pathway. Superior olivary nucleus gets descending input from cortex, sends olivocochlear fibers back to organ of Corti, inhibitory on hair cells, protects from loud sounds. Also connections to trigeminal facial motor nuclei for reflexive tensor tympanostapedius contraction.
Like built-in volume control and sound localization. Now, the vestibular system, keeping us upright and oriented.
Intrinsically linked to balanced spatial orientation. Vestibular nerve transmits info on head movement position from vestibular organs, semicircular ducts for angular acceleration, utricle saccule for linear acceleration tilt, organs in membranous labyrinth within bony labyrinth, cell bodies in vestibular, scarpas, ganglion, internal acoustic meatus. Nerve passes through meatus pontomedullary junction.
Once balance signal reaches brainstem.
Central processes terminate mainly in 4 vestibular nuclei, superior, inferior, medial, lateral, in rostral, medulla, caudal, pons. Axons leave nuclei for ascending, descending targets, integrating balance with eye movements, posture, awareness.
Ascending connections.
Axons form medial longitudinal fasciculus, MLF, preach oculomotor, trochlear, abducent nuclei, coordinates head-eye movements for visual fixation. Vestibulo-ocular reflex. Other ascending axons project to cortex via ventral posterior nucleus of thalamus for conscious spatial orientation. Axons also pass via inferior cerebellar peduncle to cerebellum for modulating equilibrium.
And ascending pathways for posture.
Axons form mediolateral vestibulospinal tracts, ipsilateral. Synapse on interneurons in anterior horn gray matter. Influence spinal motor neurons to maintain posture balance, activate extensors, inhibit flexors. This intricate system works constantly in the background, a silent sentinel ensuring we stay upright and oriented.
Moving to a small but powerful region, the hypothalamus. Tiny size, but a true neuroendocrine maestro orchestrating survival instincts.
Perfect analogy. Neuroendocrine organ, ventral most diencephalon, only 4 grams, but regulates vital processes. Fluid food consumption, temperature, sleep-wake cycle, growth, reproduction, emotion. Makes extensive connections with neuroendocrine structures. What's fascinating here is how this tiny structure integrates so many inputs to maintain homeostasis, our internal steady state. Hashtag 1. Borders and key connections, boundaries. Anteriorly by lamina terminalis, superiorly by hypothalamic sulcus, posteriorly by midbrain tegmentum, laterally by substantia innominata, rostral, posterior limb of internal capsule, caudal, forms floor-lower lateral walls of third ventricle, external ventral surface circumscribed by optic chiasm, optic tract, crus cerebri, mammillary bodies.
And its direct connection to the pituitary gland is key for its neuroendocrine role.
Absolutely crucial. Pituitary master gland, continuous inferiorly, connected by infundibulum, hypophyseal stalk, allows efficient regulation of hormones. Releasing inhibiting factors synthesized by hypothalamus pass via tubero-infundibular tract through portal vessels to anterior pituitary, adenohypophysis, controls release of ACTH, LH, FSH, TSH, GH, prolactin, etc.
So it dictates anterior pituitary release. What about posterior pituitary?
Second, distinct connection. Nerve fibers from supraoptic paraventricular nuclei extend via hypothalamo-hypophyseal tract directly to posterior pituitary, narrow hypophysis. Hormones released directly into circulation, e.g. ADH, water balance, oxytocin, bonding reproduction, allows rapid release.
This direct connection highlights its central role as a neuroendocrine powerhouse, bridging nervous and endocrine systems. Hashtag tag 2, functional divisions and key nuclei.
Internally, many small nuclei organize into mediolateral zones by fornix mamillothalamic tract and also preoptic paraventricular zones.
Let's start with the lateral zone.
Contains medial forebrain bundle, MFB, interconnecting hypothalamus with septal nuclei brainstem. Lateral nucleus in MFB promotes feeding. Tubral group connects to anterior pituitary hormone release, cerebellum motor regulation.
And the medial zone concentrated survival functions.
Yes, critical. Subdivided into supraoptic, tubral, mammillary regions. Supraoptic region, 4 nuclei for thermoregulation, osmoregulation, sleep-wake. Supraoptic paraventricular nuclei make 80-oxytocin, posterior pituitary release. Suprachiasmatic nucleus gets retinal input for circadian rhythms. Anterior nucleus regulates body temp, heat dissipation.
Highly specialized, tubral region.
Tubral region, 3 nuclei, ventromedial nucleus, largest satiety center, decreases feeding. Dorsomedial nucleus, rage, aggressive behavior expression. Arcuate nucleus, center for releasing hormones to anterior pituitary via portal system.
The mammillary region. Mammillary body, 4 nuclei, medial mammillary nucleus. Primary termination for postcomital fornix from subiculum memory connects to limbic system. Finally, periventricular zone, medial to medial zone. Neurons synthesize releasing hormones, project via tuberoinfundibular tract-to-portal system. This intricate division allows it to be a master regulator, influencing hunger, aggression, internal clock. ###3, summary of connections. From our review, it's clear this small structure has a monumental role regulating visceral, endocrine, behavioral functions through multiple complex input-output relationships with neuroendocrine structures throughout the body, constantly integrating internal-external cues. So what does this all mean for us? It means this small part of our brain silently ensures our fundamental survival, deeply influencing actions and feelings. The silent maestro. Finally, let's explore smell and how it's deeply intertwined with memories, emotions, motivation via the fascinating limbic system. A primal connection.
Olfaction plays a dual role: pleasure, food, and survival, danger. Unique sensory pathway. No thalamic relay before reaching primary olfactory cortex. Allows powerful, unfiltered connection. What's fascinating here is that direct, unfiltered pathway, likely explaining why smells are so powerfully linked to emotion and memory, often bypassing conscious processing. System begins in olfactory epithelium, nasal cavity walls, contains basal stem cells, regeneration, sustentacular cell support, olfactory receptor neurons transmission. Receptor neurons have olfactory vesicle with cilia, odorant receptors. Unmyelinated axon passes through cribriform plate, terminates in olfactory bulb. Considered CNS, embryological arch.
Considered CNS, even in the nose.
Yes. Neuron synapse with mitral cells and olfactory bulb glomeruli. Mitral cell axons converge, form olfactory tract. Divides into medial-lateral olfactory striae.
Two distinct paths.
Exactly. Medial striae, some axons septal area, others cross an anterior commissure, inhibit contralateral bulb, enhances localization. Lateral stria, primary route for conscious smell, terminate mainly in piriform cortex, primary olfactory cortex and uncus, and amygdala, emotion link. Medial forebrain bundle connects olfactory cortex with hypothalamus brainstem, regulating autonomic responses, arousal, salivation, gastric contraction.
So a direct line from nose to brain, triggering recognition and deep physiological emotional responses.
Yep.
Unique. Now, the limbic system. Intricate network of cortical-subcortical structures. Regulates complex behaviors. Memory, emotions, homeostasis, motivation. Limbic lobe. Ring-shaped cortical structures bordering brainstem. Cingulate gyrus, perihippocampal gyrus. Subcallosal area. Lateral insula also participates. Nuclear structures, amygdala, hippocampal formation, anterior mediodorsal, thalamic nuclei, septal nuclei, nucleus accumbens. This raises an important question. How do these diverse structures collaborate to generate complex phenomena like love, fear, memories, intertwining sensation, emotion, cognition? Extensive interconnectedness is key. ###3. Key limbic structures and their connections. Let's start with the amygdala. Almond-shaped, anterior to inferior horn-caudate tail in temporal lobe. 3 nuclear regions, basolateral, corticomedial, central. Function, primarily fear, also autonomic neuroendocrine pathways, fight or flight. Connections mainly bidirectional via unslated fasciculus, cortex terminalis, septal hypothalamus, ventral amygdalovagal pathway, thalamus, brainstem, forebrain.
Central to emotional responses, especially fear, nucleus accumbens.
In ventral forebrain, where putamen caudate head meet. Recognized as gratification center, role in reward addiction. Afferents from amygdala, hippocampus, fornix, basal forebrain, startura terminalis, ventral tegmentum, MFB, efferents to hypothalamus, globus pallidus, brainstem via MFB. Connection to motor system via globus pallidus influences goal-directed behavior.
Sectal nuclei.
Rostral to anterior commissure, medial hemispheres, role in pleasurable behaviors, lesions evoke displeasure rage, suggests emotional regulation role, afferents from amygdala, hippocampus, olfactory tract, brainstem, mono and menergic nuclei, widespread integration, connections to cholinergic neurons, habenular nuclei, interpeduncular nucleus, reticular formation, sleep-wake cycle.
Pleasure rage linked. Hippocampal formation, crucial for memory.
In medial ventral temporal lobe, consists of hippocampus, CA1, CA4, dentate gyrus subiculum, role in memory processes, episodic, short-term, working memory, memory consolidation. Storage likely in association primary cortices, not just here, more like processing indexing center.
How does info get into this memory center?
Input mainly from entorhinal cortex, gathers multimodal info. Projects via 2 pathways, perforant and alvear. Perforant pathway, dentate gyrus CA3 fornix, or Schaffer collaterals to CA1 fornix, or subiculum fornix, or back to entorhinal cortex, complex loop. Alvear pathway projects directly onto CA1, CA3.
And output. How do memories leave?
Efferents primarily from subiculum form the fornix, arch. White matter structure arches over ventricular system, begins as alveus fimbria, emerges, curves under corpus callosum. Divides at anterior commissure into pre-postcommural fornix. Targets: nucleus accumbens, septal nuclei, medial frontal cortex, mammillary nucleus, ventromedial hypothalamus, anterior nucleus of thalamus.
All ties into the classic Papez circuit.
Indeed. James Papez, 1930s, describes circuit linking these structures cortical areas thought involved in emotion. Pathway: subiculum fornix, mammillary nuclei, mammillothalamic tract, anterior nucleus of thalamus, internal capsule, cingulate gyrus, cingulum fibers, parahippocampal gyrus, entorhinal cortex, hippocampal formation. Modern view. While useful for pathways, some structures' roles in emotion less direct multifunctional.
The Papez circuit is a classic example of evolving science. Even if the emotion part isn't as clear-cut, it still illuminates a profound network for memory and broader limbic functions. Wow, what an incredible journey we've taken today. From the earliest spark of embryonic development to the intricate structures of the brain and spinal cord, the specialized pathways for vision, hearing, balance, right down to the fundamental orchestrations of the hypothalamus and the complex interplay of the limbic system. So what does this all mean? It means the more we uncover, the more we appreciate the awe-inspiring intricacy, resilience, and elegant design of the human nervous system.
Absolutely. Mastering these details isn't just about memorizing names, it's about appreciating the profound functional elegance that defines our very existence. Each structure, each pathway is a testament to evolution, enabling everything from simple reflexes to complex thoughts and deep emotions. If we connect this to the bigger picture, mastering these details isn't just about memorizing names, it's about appreciating the functional elegance that defines our very existence. Gaining this elite professional level understanding truly transforms how you view yourself and others, and how you approach health, disease, and what it means to be human.
And as we close out this deep dive, consider this provocative thought. Given the sheer number of interconnected systems we've explored and the cascading effects of even small changes, what does this elaborate neural architecture tell us about the future potential of advanced brain-computer interfaces or truly effective treatments for complex neurological disorders? How much closer are we to truly replicating or repairing such a magnificent system? Thank you for joining us on this deep dive.